Humans have known breeding techniques since a long time. They have been using these breeding techniques with animals and they have been picking up and choosing traits which they seem desirable to get the desirable animal. Hello everyone, my name is Ekta and today we'll be talking about BT cotton. With the knowledge and tools of genetic engineering, scientists can now tweak and alter the genetic code of organisms and yield desirable results. What is BT cotton? BT cotton is a transgenic plant which is able to prop up a defense against pests that infect it. Pests like Lepidopteran larvae, for example, moth, butterfly, beetle, and bollworm. Genetically modified crops are produced by applying the knowledge and techniques of biotechnology. BT cotton is able to produce an insecticide to tackle its problem of pests. BT cotton's creation eliminates the need to use broad spectrum insecticides. The story of BT cotton begins with the discovery of BT, Bacillus thuringiensis. BT refers to Bacillus thuringiensis, which is a soil bacterium that was identified as an insecticide. So it was able to kill certain specific insects. It was discovered by Shigetane Ishibatari as the cause of sudden collapse disease of silkworms in 1901. It produces crystals of endotoxin, cryprotein, which acts as an insecticide to worms like bollworms in their larval stages. Strains of bacteria produce variety of Bt toxins. Cry genes are expressed mainly during the stationary phase of bacterial sporulation. Cry1AC and Cry2AB are types of genes that control bollworms. Now you may think, what kind of protein is cryprotein? So cryprotein acts as a stomach protein for the larvae of moth, beetle and butterfly. So on ingestion it basically poisons these insects and it acts on them so that they die. Upon investigating this cryprotein, we found out that these are fatal for the larvae when they eat it. So, on ingesting this cryprotein, the larvae stop eating within 2 or 3 hours and then they die subsequently. Bt crystals are actually the proteins. So, the proteins are in the form of crystals, which are referred to as ICP, insecticidal crystal proteins. So they are a type of proteins. As you can see in this diagram, some kind of crystal structures are visible. They are formed during the sporulation in bacteria in stationary phase and are coded by the cry gene. So the cry gene is the gene responsible for the coding of this cry protein. Now let us look at the mechanism of action of Bt toxin. How does this toxin work when it is ingested and it goes inside the body of the worm? So the mechanism of action certainly begins with the ingestion of this toxin by the insect. The crystal proteins undergo activation in the insect gut under alkaline pH. So when they enter, there is a basic pH inside the body or the gut of the worm, and there this toxin gets activated under the gut proteases and they become active toxins. These active toxin proteins bind specifically to brush border membrane vesicles, and this is present in the midgut epithelium cells. So, in the midgut, the active toxin proteins bind to these membrane vesicles of the epithelium cells. This creates pores in the midgut. So, this leads to creation of holes or pores in the midgut, and these pores are non selective. So, they are quite permeable and ions and small molecules can pass through them. Now, because of this, the osmotic lysis of the membrane occurs, which leads to septicemia and eventual starvation and death of the insect. After the identification of such a gene, which can specifically cause the death of these insects, which were a great cause of pain and devastation of cotton yield. So, the next possible or the next effective step was to certainly use the genetic engineering, which was at the scientist's disposal to create GMOs. Now, what is the basic idea behind any biotechnology venture? Is to simply apply genetic engineering techniques to get this endotoxin producing gene incorporated into the cotton cells. That is what is presented in this diagram that you can see. That the gene which is responsible for the endotoxin is getting incorporated into the cotton cell. And now, after getting incorporated into the cotton cell, the cotton will be able to produce this endotoxin on its own. And when it will be able to produce this endotoxin, the cotton bollworms, including other worms, will not be a botheration to the cotton plant. There are several steps that are involved in the production of any GMO like the BT cotton. Some of these general steps are given here in this diagram. So let us go through them one by one. First is the identification of a trait of interest. So here in Bacillus thuringiensis, the trait of insecticidal properties that was identified was the trait of interest. These were ideal for a plant like cotton, which loses a lot of yield due to the attack of these insects. So that was the trait. Now the second step is that for this trait, which gene is responsible, the scientists have to identify. So the desired gene has to be isolated. And there are a lot of applications and a lot of softwares at the disposal of scientists to identify which gene is responsible. Now, also, there is a lot of genomic analysis which goes behind it, When the gene is identified, when the sequence is identified, then restriction digestion is done and the gene is separated from the genome. The gene is then amplified using PCR. And then the third step is that this gene, which has now been separated and amplified, is ligated to a plasmid vector or a carrier. After the ligation, we have a plasmid which has the sequence of gene of interest. Now, this recombinant DNA, as we call it, is inserted in our seed, which will grow up to be the cotton plant. Now, there are a lot of techniques which are used to insert or infect the cells. It can be done by gene gun or it can be done by a plasmid infection. Then the last step is sowing these seeds and giving them controlled, careful monitor, and then these seeds grow up to be the GMOs with the genetic expression which expresses the endotoxin and is resistant to pests. Now let us quickly look at some of the advantages of BT cotton. There is a significant yield loss due to bollworms. So here BT cotton protects against three types of bollworms: American bollworm, Helicoverpa armigra, then pink bollworm, Pectinophora gossipila, then spotted bollworm, Aries vitilla. Insecticide worth 704 crores were used for bollworm control in 2001. So there was a lot of money spent on insecticides, and hence BT cotton, which already is at the disposal of an insecticide within itself, can produce and manufacture insecticide within itself, does not need insecticides from outside. So there is a lot of money saving there. And then these are environmentally friendly. That means if any other organism ingests this, it will not die or there will not be any adverse effects. These are the three bollworms which infect. There is the spotted bollworm, then the pink bollworm in the middle, and then the Helicoverpa. Now, commercialization of BT cotton. That meant which firm or which company was the one leading the way with the BT cotton. So there was a company known as the Monsanto. So Monsanto was a company based out of US which introduced the BT cotton in India. And after its introduction in 2014, it was discovered that 95% of cotton in India was BT cotton. India today is the largest producer of cotton and also the largest producer of BT cotton in the entire world, which is closely followed by China. There's a lot of debate on what are the benefits and what are the risks of GMO. So let us read together. Nutritional value of food could be improved is a benefit of GMO. However, a risk can be new traits can cause adverse health reactions. Then crops can be produced that lack known allergens. and removal of traits have unknown effects. Crops can grow in arid regions so they can have a better yield, and crops may limit biodiversity of a local environment. Another benefit can be that GM crops can produce herbicide to kill pests. Then risk can be cross pollination can lead to superweeds. Then another benefit can be GM crop may have longer shelf life. And another risk can be food with GM components may not be labeled. So as you can see, there are a lot of benefits, and equally there are certain risks of GMO. Because when we genetically modify an organism, the outcomes that can be there or the adverse reactions are or cannot be predicted in future. So all of these adverse reactions are only seen once their population is there in there for everyone to use. So it's almost like a medicine, that a medicine adverse reaction can only be known once it is tested. So there is a lot of hype, or a lot of negative hype, I would say, among a lot of people regarding and concerning GMOs, and there are a lot of ethical issues surrounding it as well. I would like for all of you to search about the ethical issues regarding the GMOs. Why is it considered in some communities unethical? So now there is a short exercise which you all can do regarding what all you've learned here in this video. The first question is, what is the mechanism of action of Bt toxin? The second question is, what are the main steps in production of a GMO? The third question is, what are some of the pests of cotton plant? And fourth question is, you have to list advantages of Bt cotton. If you will do this exercise, you'll be able to recapitulate better and you'll be able to retain better also. Thank you all.